To create a new human, biology has to solve a math problem. It must take a complex 46-chromosome diploid genome, split it perfectly in half into a haploid state, and then fuse it with a second haploid cell to restore the original chromosome count. We reproduce sexually. We are viviparous, meaning we give live birth. This requires the male and female systems to execute a series of anatomical and chemical events with extreme spatial and temporal coordination. As you can see in these typical exam questions covering spermatogenesis, ovulation, and IVF, mastering this sequence is a strict requirement in advanced biology and pre-medical studies. We are dealing with precise, testable mechanisms. The timeline begins with gametogenesis, the manufacturing phase where the raw biological materials, sperm and eggs, are assembled. Once the materials exist, the female body initiates a neuroendocrine preparation sequence known as the menstrual cycle. This sets a very narrow window for the next critical event, fertilization. If fertilization succeeds, the timeline shifts into embryonic development, culminating 9 months later in the neuroendocrine cascade of parturition, or childbirth. The system operates through the total integration of structural anatomy and neuroendocrine timing, where precise chemical signals drive every physical phase of the cycle. Gametogenesis is the actual manufacturing phase, taking place within the primary sex organs, the testes in males and the ovaries in females. In males, spermatogenesis begins at puberty and operates like a continuous assembly line. Once it starts, it never stops. This flowchart tracks the process deep within the seminiferous tubules. A single primary spermatocyte undergoes meiotic division into 2 secondary spermatocytes, dividing again into 4 haploid spermatids. Through spermiogenesis, these round spermatids transform, shedding excess cytoplasm to develop the structures needed to swim. The resulting spermatozoon is highly specialized for one task. The head contains the genetic payload and an enzyme-packed cap called the acrosome. Behind it, a densely packed ring of mitochondria powers the flagellum, or tail, driving the sperm rapidly forward. The female system handles gamete production entirely differently. Oogenesis does not begin at puberty. It initiates while the female is still a developing embryo in the womb. This flowchart compares the systems. Spermatogenesis continuously creates 4 equal cells. Oogenesis remains dormant for decades, completing division sequentially. Notice the extreme asymmetry. Meiosis produces one massive, nutrient-rich ovum, discarding leftover genetic material into non-functional polar bodies. The evolutionary strategy is clear. Male gamete production optimizes for sheer quantity and motility, sending millions of tiny messengers. Female gamete production sacrifices quantity to build a single, perfectly resourced target. To prepare for that target, the female reproductive system runs a roughly 28-day countdown sequence, driven by the hypothalamic-pituitary-gonadal axis. This graph plots the cycle. Rising FSH triggers follicle growth. It secretes estrogen, thickening the endometrium. Around day 14, an LH surge triggers ovulation. The ruptured follicle becomes the corpus luteum, releasing progesterone to hold the uterine wall at maximum thickness, awaiting a fertilized egg. The menstrual cycle operates as a highly calibrated chemical timer. It constructs a temporary biological landing zone precisely at the moment the ovum is released. For fertilization to happen, the sperm and egg must meet at a specific anatomical bottleneck, the ampullary isthmic junction of the fallopian tube. Reaching the egg, the sperm executes the acrosomal reaction. It releases specialized enzymes to dissolve a pathway through the ovum's thick outer layer. These enzymes allow the sperm to physically penetrate the protective barrier, known as the zona pellucida. The exact moment the sperm membrane fuses with the egg, the ovum initiates the cortical reaction. The egg's membrane instantly hardens, blocking the entry of any additional sperm to prevent a fatal condition called polyspermy. The entry of this single sperm triggers the ovum to finally complete its second meiotic division. The 2 haploid nuclei fuse, producing a single diploid cell, the zygote. This illustrates cleavage. Traveling down the fallopian tube, the zygote undergoes rapid mitotic divisions, 2, 4, then 8 cells, without increasing total volume. Reaching the 16-cell morula stage, it migrates to the uterus. By day 10, the hollow blastocyst embeds securely into the prepared uterine wall. Fertilization represents a strict sequence of biochemical triggers followed by a precise physical migration. Post-implantation, the developing embryo and the maternal tissue form the placenta. This structure acts as a physical life support bridge and a massive temporary endocrine organ. To maintain the pregnancy, the placenta secretes hormones found nowhere else in the body, including human chorionic gonadotropin, human placental lactogen, and high levels of progestogens. At the end of the 9-month gestation period, the system must reverse its priorities to expel the fully grown fetus. This initiates parturition, a complex neuroendocrine process. The trigger comes from within. The fully developed fetus and the placenta generate the initial signals for what is known as the fetal ejection reflex. This reflex causes the uterine muscles to begin mild contractions. These contractions send a neural signal straight to the mother's pituitary gland. This circular flowchart illustrates the resulting positive feedback loop. The pituitary gland releases oxytocin, causing the uterus to contract. This cycle rapidly amplifies. Stronger contractions force more oxytocin release, which forces even harder contractions, escalating until the baby is delivered. Childbirth relies on temporarily hijacking the body's normal homeostasis. It utilizes a self-amplifying chemical loop to achieve a definitive physical output. Watching the timeline reverse, delivery retracts, the blastocyst detaches, and the zygote divides back into an egg and sperm. The dependency of each step becomes obvious. Human reproduction demands absolute harmony. Spatial anatomy must perfectly align with temporal endocrinology. Whether it is the microscopic precision of the acrosomal reaction or the macroscopic, systemic scale of the oxytocin loop, every mechanism is part of a single, continuous system. It is a process that successfully transforms two highly specialized, extremely vulnerable haploid cells into a fully differentiated, independent human organism. It stands as an incredible feat of biological engineering, a system designed to perpetuate the species by flawlessly executing a millions of years old chemical blueprint.